The overall goal of this procedure is to quantitatively investigate the metabolism of multiple nutrient sources using labeled substrates. This workflow allows the fate of consumed nutrients and the metabolic origin of compounds synthesized by yeasts to be determined.
This method can help to answer key questions in the microbial metabolism field, such as how the metabolism operates depending on environmental conditions. The main advantage of this technique is that it allows the elucidation and quantification of the pattern of nutrient distribution from multiple sources.
This method was used to investigate the nitrogen metabolism of yeast, but it can also be applied to other metabolisms and microorganisms. The key to success with this experiment lies in the reproducibility of fermentations. Users must pay a particular attention to collect the cells in the same defined physiological stage. Demonstrating the procedure will be Picou from our technical staff, Pauline Seguinot, a PhD student from the laboratory, and Rollero, a postdoctoral fellow.
In an autoclave, pasteurize each synthetic medium prepared for each nitrogen source in a 1-liter flask containing a magnetic stir bar. Dissolve the appropriate amount of labeled molecule to reach the desired final concentration in the medium. Sterilize the medium using a disposable vacuum filtration system. Using a sterile measuring cylinder, divide the medium between 2 pre-sterilized fermenters that contain a magnetic stir bar and are equipped with fermentation locks to avoid the entry of oxygen but allow the release of carbon dioxide.